After the larval stages and at the beginning of puparation, the disc eversion takes place. So let's remember first what's the structure of the imaginal disc. It is composed of two layers, one which is the proper disc, which is a columnar epithelium, as you can see here, and a layer of squamous epithelium, which is the peripodial membrane, which does not give rise to any adult tissue. And this disc is attached to the larval epithelium through the stalk, which is this region here. So here you can see a frontal view of the disc, and in B to D a lateral view of the disc. During the process of eversion, the disc first contracts and then it goes outside in this way, and in the end the disc is between the larval epithelium and the cuticle. So remember that at this stage larval epithelium and cuticle have become separated by the process of apolysis, and this cuticle is indeed the pupae. The eversion of all Drosophila discs is roughly synchronized, and it takes place after the ecdysone peak that triggers that starts puparation. So here you can see two imaginal discs of the two legs that have been dissected and cultured, and ecdysone hormone has been added. So this ecdysone can trigger disc eversion, leg disc eversion in vitro, and the same happens with the wing disc. Here's the process of eversion from outside, like from a view that is outside the larval epithelium. This gray shade is the larval epithelium. Here you have the stalk, and the red part would be the peripodial membrane, and the yellow part is the proper disc. First of all, the peripodial membrane expands and surrounds the proper disc. Then holes start forming in the larval epithelium close to the stalk region, and later these holes join in a bigger hole, and the notum everts through this hole, and after the notum the wing blade everts. So during this process also takes place the folding of the dorsal and ventral part of the pouch, so that they get opposed to each other, and the disc goes from being a flat epithelium to be a two-layer epithelium, the proper disc. Okay, so the ventral and the dorsal layers get opposed to each other, and this process takes place through basal constriction. So here you have the disc in a lateral view. The gray part is the proper disc, then the green part is the peripodial membrane, and the dark green is a myosin band that is in the peripodial membrane and that starts contracting. So here, as you see in figure B, this folding has already taken place through basal constriction, and therefore the dorsal and the ventral parts of the pouch are opposed to each other, and they start elongating a little bit. At the same time, this myosin band contracts. Therefore, because of these two processes, the peripodial membrane cells here are under a lot of tension, and therefore the disc folds like this. It's because of the pressure of the peripodial membrane, which the disc folds. Here you see how the myosin band contracts more and more, and in the end here you see how this hole is the stalk, and this is the hole that will be used by the notum to emerge. Here you see the same thing, but in real pictures. In green, myosin is labile, So as you can see, myosin contracts, myosin bands contract here, and then a hole forms and the wing disc emerges through this hole. Here you can see a video. Now the wing disc is emerging from the peripodial membrane. Okay, let's see it again. The myosin contracts, and finally first the notum and then the blade part emerges, and immediately the blade starts expanding. Okay, so here you see the same thing. Here's just to show the myosin band, which can be visualized here at the lateral part of the peripodial membrane. Okay. So if you downregulate this myosin, for instance, by downregulating CYP1 or CYP, which is the light chain of the myosin, you can see that compared to this control, this was control. Well, eversion does not take place. Okay, in this video, the disc is not able to avert. Let's see the tissue in more detail. Here you have semi-thin slides, and the pink region here is the peripodial membrane, the yellow region is the proper disc, and this greenish layer is the larval epidermis. So as you can see here, both the disc and the larval epidermis are separated in larval stage 3, and they are both surrounded by a basal membrane, which is marked by red arrows, and you can see as a black line surrounding the tissues. However, 3 hours after pupae, this pink region of the peripodial membrane gets very close to the green region, which is the larval epidermis. And as you can see in this electron micrograph, the basal membrane disappears between both tissues. So when eversion is going to take place, this region that is close to the stalk, this where the yellow arrow is, would be the stalk. So in this region of the larval epidermis, some cells from the peripodial membrane start to intercalate and to emerge, which would be these green cells. Okay, so the cells of the peripodial membrane start integrating into this larval epithelium. Some holes form, and the lumen of the disc starts to be in contact with the space that is between the cuticle and the larval epidermis, as you see here, and also here. Okay, here you see some of these holes that form as a result of this intercalation of cells from the peripodial membrane into the epidermis. And in the end, all these holes are going to join, and as you can see here, okay, here in this video, the hole is forming and increasing its size. Different holes join, and finally the disc eversion takes place. Additionally, usually until this moment, cells that are clonally related, which is cells that come from one cell in at some point, usually are very tightly packed together, like in this black clone here. However, these cells that are at the front, the ones that start this eversion process in the peripodial membrane, although they are closely related, they lose contact with each other, like you see in these blue cells here. In this in this drawing, it's marked which cells are they. So these cells in the peripodial membrane lose contact with each other, and they start generating these lamellipodia and also philopodia, and they start moving a lot. Okay, so they undergo what is called an epithelial to mesenchymal transition. Also, they lose in the process of losing contact with each other, they start relocalizing the components of sonula adherens and septate junctions, which are the most important cell junctions types that there are. Sonula adherens are in the apical part, and they have also a signaling function, and septate junctions are in the more lateral part, and they prevent solutes or ions to move from the apical to the basal side, and vice versa. So here they label in red the cadherin and armadillo, which are two components of sonula adherens, and they see how, whereas in the disc they are normally in the cell-to-cell junctions here in red, in this green part, which is the one that is undergoing this transition, they stop being located at the cell junctions. Okay, they just disappear from the cell junctions. Here you can see from the lateral side the location of armadillo. And here they show the same thing for a component of the septate junctions, which is coracle. So these cells will go a true epithelial to mesenchymal transition because they lose contact with each other, and they change the location of these proteins. The loss of these components from the apical surface and also the upregulation of cell motility is regulated by the GNK pathway. So if this pathway is downregulated, this epithelial to mesenchymal transition does not take place, and cells don't start the movement, and this does not develop properly. But if the pathway is too upregulated, the cells move too much. This pathway has to be tightly regulated. And finally, in this video, it is shown how the cells that of the notum, that are the ones that emerge first, migrate to the dorsal midline and get in touch with the contralateral cells of the notum. So they form the external part of the thorax.